All right, thank you, everyone. Okay, every year, 33 billion pounds of plastic enter the ocean. That's roughly the equivalent of 3 truckloads full of plastic entering the ocean every minute. And this problem is expected to intensify as plastic production is set to nearly triple by 2060. And of course, once this plastic enters the ocean, it doesn't just disappear. It breaks down and becomes nano and microplastics. It's estimated there are over 170 trillion of these particles in the ocean right now. That's enough for every single living person on Earth to have 20,000 plastic particles. And Hawaii is especially susceptible to this plastic debris because it sits in the middle of the North Pacific Gyre. The islands act as a filter and they catch the marine debris that's carried around by surrounding currents. So between the ocean currents and local inputs, Hawaii's reefs are very heavily impacted by plastic debris. And so when we think about a solution to the plastic pollution problem in marine systems, there are really 2 ways to tackle the problem. The first one is dealing with the plastic that's already in the ocean. And the second one is making better materials so we don't keep adding problems as we move forward. So starting with that first challenge, dealing with plastic that's already in the environment, I'm focusing on how microbes interact with and potentially degrade polyethylene terephthalate and PET, and PET-associated compounds in Hawaiian reef systems. So the workflow for this is pretty clear. We start out by gathering samples from reefs impacted by plastic. We isolate microbes that can grow on PET as the sole carbon source. We screen for PET-related traits, and then we functionally validate PET degradation. So our first step is we gotta find those microbes that can live off of PET as their only carbon source. But first, why PET? Well, PET is incredibly prevalent in reef systems, in the sediment and water, and it's most frequently found within coral tissues over all other plastics. Importantly, compared to other common plastics like polyethylene or polypropylene, PET is more accessible to microbes. It has these ester bonds holding it together that can be hydrolyzed. And when these ester bonds are cleaved, they produce byproducts that are relatively easy to separate and therefore to quantify. So the first step is I get to go to my lovely field site on Sand Island, and this is where I know there's going to be lots of reefs with a lot of plastic pollution. I bring these samples back to the lab and I grind them up in a blender to make coral smoothies. And this way I'm getting microbes extracted from the skeleton, the tissue, and the mucus all at once. The first problem I ran out against was trying to get the very hydrophobic and static PET into the agar. So I knew that PET has a glass transition temperature of around 70 degrees. That's when the polymer gets very flexible as opposed to rigid. And I knew that it doesn't degrade at any temperature that's higher than 121 degrees, which is what the autoclave is at. So I took this plastic and I put it in some agar and I stuck it in the autoclave. And what we have in the horizontal or the vertical picture is the PET before the autoclave, hydrophobic and not mixing. After the autoclave, it went in nice and we have our nice dispersed plastic in the agar. But if you ever try this, you will end up with a big plastic chunk on the top, and you will have to spend about 15 to 30 minutes fishing that out of the bottle, just to warn you. So then, from the coral samples, I isolated 10 distinct microbes that have been sustained on PET as their only carbon source now for 7 months. These isolates span multiple genera, including taxa previously associated with plastic degradation, and genera with no documented PET activity. Step 2, we have to test to see if these microbes have the enzymes necessary to break down PET bonds. So here is our PET polymer, and when it breaks down, it's converted into smaller intermediates, BHET and MHET, and those are further broken down into the monomers TPA and ethylene glycol. So we're going to focus on the intermediate BHET. BHET is a smaller and more accessible polymer than PET, but requires the same enzymes to degrade. So it serves as a useful model substrate. Therefore, we can screen our isolates on BHET hydrolysis, or for BHET hydrolysis using a clearing assay as a proxy for PET degradation. So here's how this looks. We have our plate over here with a PHET, and what we want is for the microbes to degrade it until the plate becomes clear again. Here's the published example of this going on. You can see the darker circle around the hyphae of the fungi is actually where the fungi is degrading the BHET. So that's what we're looking is for those clear zones. And here are two examples of two of my isolates that are able to degrade BHET. So on the top row, we have a very strong degrader. You can see that the opaque region of this plate is getting smaller and smaller until at 4 months it completely disappears. So this guy was able to completely degrade the BHET polymer. At the bottom, we have a very slow degrader. This is our clear slime. And for 3 months, I thought this guy was unable to degrade BHET until a few weeks ago when it started to degrade it pretty quickly. So I'm not really sure what's going on there, but we do have multiple isolates now that degrade BHET. So that means they have the enzymes necessary to break down PET plastic. This is a picture of what I looked like for weeks after discovering this. Thank you, Iker. Okay, step 3 is testing for PET surface colonization. So what I did is I took broth with no carbon source, and I put into the broth as the only carbon source the PET film. And I was thrilled that after a few weeks, I actually started seeing these biofilm-like structures forming on the PET. And this means that the PET can overcome the hydrophobic barrier of plastic. And this is a key step for the enzymes to be able to make contact with the plastic and start degrading it. I also did this with PET powder, 16 microns. And if you look at the control compared to the treatment, you can see that the treatment actually has aggregated PET particles. So it looks like biofilm structures there. So what we know so far, we know that our isolates demonstrate that they can utilize PET and PET-derived compounds as a carbon source. They exhibit enzymatic activity, and they can interact with PET hydrophobic surfaces. And so here are our stars of breaking down BHET and producing biofilm on both the film and the powder. Then we have our second place guys. These guys can break down the BHET and form a biofilm structure on either the film or the powder. In third place, we have these guys who can break down BHET but have formed no structures yet. And then lastly, we have yellow slime, which looks really cool, but doesn't do anything but actually live off the PET compounds. So taken together, these results point toward a functional interaction between coral-associated fungi and PET-derived compounds. This suggests that these fungi may be participating in PET degradation in reef environments. Future directions is to validate the PET degradation. So we're currently in the process of taking those inoculations in the tubes and putting them through the HPLC to quantify PET degradation products. We're going to validate chemical bond transformation through FTIR, and we're going to visualize physical surface erosion through SEM. So stay tuned for that. And so stepping back a little bit, one potential long-term application of this work, I think, is the development of beneficial microbes to have a consortium that can be used to enhance coral resilience to plastic-associated stress. So kind of a plastic-fighting coral probiotic. And I think my contribution to this could be to identify and functionally validate candidate microbes for this probiotic in Hawaiian coral reef systems. So basically, I kind of want to find the ingredients for this probiotic recipe. And the types of functions I'm interested in are finding microbes that can break down plastic leachate, such as plasticizers and flame retardants, microbes that can fight off pathogens that are commonly found on plastic debris, microbes that can neutralize Ross species, because when corals have in contact with plastic, they actually produce Ross species. And also looking for microbes that can support symbionts, because plastic also decreases photosynthetic efficiency. So one way I've been doing this is I'm starting to look at microbes that can neutralize Ross within the Hawaiian corals. This assay uses DPPH, which is a stable free radical that appears purple in its oxidized form. But when DPPH is reduced by antioxidants, it loses its color and becomes this pale yellow or white color. So in this setup, I grew isolates on filter paper, which I then pipetted TPPH onto. And fungi that produce extracellular antioxidants reduce the DPPH in the surrounding area, resulting in white halos. So what you're seeing here are isolates that can be relevant for mitigating oxidative stress caused by plastic debris in coral environments. Okay, so that's the first part of the solution. But the second part of this approach is addressing the plastic pollution material or plastic materials. We want to find materials that can actually degrade in the environment to replace the current plastics we have. So as part of this goal, I'm testing the degradation abilities of newly developed a TPU, thermopolyurethane plastic. TPU is widely used in the hospitals, in consumer and industrial sectors, in shoes, equipment for sports, all sorts of things, and it doesn't degrade easily in the environment. So Dr. Amon's lab worked with the University of Notre Dame, and they actually came up with an idea of putting a polysaccharide inside of a TPU polymer. And they're hoping that the incorporation of this very labile carbon source can stimulate microbial activity and enhance interaction with the polymer. So Dr. Yu Xiao at the University of Notre Dame made this TPU plastic, and I'm going to test to see if microbes can degrade it. So because TPU does not break down in easily measurable products like PET, I had to develop a new pipeline to test degradation. So what I did is first, I wanted to test metabolic activity with resveratrol. Resveratrol is reduced during cellular respiration, so you expect a color change from purple to pink if the fungi were metabolically active. And so I plan to use this signal as a proxy for their ability to metabolize carbon from the TPU. The next metric I was going to use was weight loss. Of course, you want the weight of the polymer to decrease after being incubated with the microbes, because that means that some of the plastic was degraded. And then we're going to look at the surface erosion through SEM imaging. So here's the experimental setup. We have each yellow block is a different fungal species. We use Dr. Amon's infidel degrading fungi for this project. And then each column is a new treatment with 4 replicates per species. So this guy right here is the experimental. It has the TPU and the fungi. Then we have our negative control. We have fungi with no TPU. Positive, just glucose and fungi. And the sham control with heat-killed cells and TPU. And by day 4, I knew that we had a problem because the experimental was being reduced, but also the negative control was being reduced. And what we reckoned is that there was probably metabolic activity driven by carryover carbon reserves. So probably these guys had carbon in their stored inside of them, and that's what was reducing the resveratrol. And 3 weeks later, we had another problem. We had this yellow discoloration, and it looked like all of the wells were being reduced. So we realized that resveratrol becomes unstable over time, and it's not viable for a long-term assay. So that metric was out. Next metric was weight loss. And so I washed and weighed and dried all of these plastic pieces. And after having the before and after weights, I realized that the mass had actually been gained by the polymers. This is probably because this TPU is very porous, so it probably retained the water in the biofilm. So that metric was also out. So our last metric to use was SCM imaging. And Dr. Rivers got some really great images of the fungi interacting with the TPU. Here is an example from a publication that shows how this should work. So the before picture should be nice, smooth plastic, and the after picture is actually showing where the microbes degrade the plastic. Unfortunately, this is our results. Our before pictures were very rocky and not uniform, and so you couldn't really tell them apart from the after pictures. And so we had to get rid of this metric as well. So this means that we have a redo to do on this experiment. And the first thing I wanted to do was test to see if the resveratrol assay could really be used to tell us about plastic degradation. We knew that the resveratrol assay only lasts a few days, so the cells cannot be incubated in it. And we knew that it will give a false signal if there's carryover carbon reserves. So I wanted to ask these 2 questions. First, if I take the cells away from their carbon source and put them in a carbon-free assay, can they still reduce resveratrol? And second, can I starve them enough to remove any carryover carbon so the signal actually reflects what I want it to reflect? And so to do this, I incubated the cells in either a carbon-free broth or a rich MEA broth, and I transferred them to these resveratrol assays that were 24 hours, and I did it over a week. And here are the results. On the x-axis, we have the days. I'm showing days 1, 3, and 6. The y-axis, we have the change in the resveratrol signal over the 24 hours. And the red line is our positive control. So here are the cells that were removed from the carbon source and then put into the no carbon resveratrol as assay. And they actually reduced the razazirin away from their carbon source. So that was a success. We knew we can take things away from their carbon source and they'll still reduce the razazirin. The purple line is the cells that were starved. And so we showed that if we incubate these cells with no carbon, they can be starved and they won't give us a false signal. The problem is this blue line hiding behind the purple line. That one is starved cells that were put into razazirin and then given a very labile carbon source. And the problem is they never really came back. And so this introduces a lot of ambiguity in interpreting these results. And in order to use this assay, we really need a lot more work to figure out exactly how to use it. But I was able to get more uniform plastics from Dr. Shao, so we can now use SEM for this metric. And I was given this information from Dr. Shao about his TPU. This tells us that his plastic can be pulled and stretched from 3 to 8 times longer, and it will until it breaks. So this means that we can use instron testing, which quantifies stiffness, strength, and elongation. So we have this metric, SEM, and we're going to use FTIR and Raman, which look at change in functional groups and molecular structure and bonding. And we're starting over by actually taking samples from the environment to try to find any microbes, bacteria, or fungal that can break down the TPU. And we're also doing antagonism studies to see if we can put together a consortium of the imprenel degrading fungi that Dr. Amond already has and see how well they can degrade the TPU. And so far, our results have been really great. In the blue, you see our control. In the orange, you see our bacteria with quite a bit of biofilm. And in the middle, we see fungi with a ton of biofilm. I've never seen that much biofilm on plastic before. So it looks like the microbes are really liking this altered TPU polymer. And I just want to say thank you to Dr. Amond and the Amond Hansen Labs. I'm not a microbiologist, and you've all taken time to answer questions and help me, and you've been so generous. So thank you very much.